Hi students, today we are starting our next topic of the chapter, that is types of RNA. Three types of RNAs are there: messenger RNA, ribosomal RNA and transfer RNA. As we know, messenger RNA is responsible for taking the messages. Ribosomal RNA, it is present on ribosome, and transfer RNA, it transfers the messages from one place to another. Messenger RNA, it is synthesized inside the nucleus. It works as a complementary strand of DNA and it provides the template. Template means it gives the code. It constitutes 10% of total RNA present in the cell and it has lifespan of about 2 minutes in the bacteria. Example: monocistronic messenger RNA and polycistronic messenger RNA, means which has single stranded structure, that is known as the monocistronic messenger RNA, which has more than one strand, that is known as polycistronic. Ribosomal RNA, it is present in the ribosomes, which are nucleoprotein molecules, 80% weight of total RNA of the cell, and it plays catalytic role. As we know, catalysts are the substances which are never taking part in the reaction, but they increases the rate of the reaction. Example: 28S and 18S. Transfer RNA, it is formed of 60 small sized ribonucleic acids. It brings amino acids and reads genetic code. 10 to 15% of total weight of RNA of the cell. Example: 4S. As we can see, this is the messenger RNA. This is adenine, guanine, uracil. Now, ribosomal RNA, it is present on ribosome and which brings amino acid with it. It is known as the transfer RNA, as you can see on the screen. Now, we will start with polymerization. It takes place in following three steps, that is initiation, elongation and termination. Initiation means starting of the reaction. Elongation means it gives length to the reaction, and termination, it stops the reaction, initiation. RNA polymerase binds to promoter and initiation starts. This is the process when polymerization process starts, and it uses nucleoside triphosphate as substrate. Elongation. RNA polymerase is only capable of catalyzing the process of elongation. It associates quickly with the initiation factor, that is the sigma factor, and termination factor, that is known as the rho factor, to initiate and terminate the transcription respectively. Termination. Once the polymerase reaches the terminator region, RNA falls off because of RNA polymerase, and this results in termination of transcription. Now we will see initiation. As you can see, this is the sigma factor attaches to the DNA molecule. This is known as the initiation reaction. As sigma factor reaches to the almost center of the reaction, that is known as the elongation of the reaction, and it is when it is reaching to the end of the reaction, then sigma factor converts into rho factor, which shows the end of the reaction, and at the terminator end, RNA polymerase and rho factor is released. So during polymerization, three important reactions takes place, that is initiation, elongation and termination. As we can see, you can see here, it is the DNA molecule promoter. This is RNA polymerase reaction. Now here coding changes. This is the direction of the synthesis. As we know, adenine is replaced by thymine, guanine by cytosine. As you can see, this is the non-template strand. Non-template strand means this is the strand where coding is written but it is not converted yet. Now here you can see thymine converts to adenine, adenine converts to thymine, cytosine to guanine, thymine to adenine, guanine to cytosine, cytosine to guanine, and like that. And this is new formed strand is known as the template strand. Today whatever I have taught you, I am sending you its PDF. Thank you.